We're going to move to our next speaker, Megan Gliozzi from the U.S. Food and Drug Administration.
Good afternoon. I'd like to thank the organizing committee first for giving me this opportunity to present some of my research. I'm a postdoc in Dr. Heather Painter's lab, where we focus primarily on understanding transcriptional dynamics across the blood stages of Plasmodium falciparum. This particular project focuses on the interactome throughout these stages. Next slide. It's becoming more apparent with continued research that post-transcriptional regulation plays a major role in stage-specific gene regulation. This is partially evidenced by the discordance in mRNA and protein abundance peaks throughout development. But also, if you think about it from an evolutionary standpoint, we know of only 27 transcription factors that regulate transcription across the entire genome throughout all of development. On the other side of that, there's an estimation of up to 1,000 RNA-binding proteins, or RBPs, dedicated to post-transcriptional regulation. And so just by the numbers, it seems that evolutionarily, the odds are stacked towards post-transcriptional regulation, making some of these RBPs likely critical for life cycle progression and also possibly promising drug targets. However, the RNA interactome in Plasmodium falciparum is largely still uncharacterized, mostly due to a lack of effective methodologies in vivo. We'd like to step in here to introduce a novel method for Plasmodium called parCL to get a better understanding of the interactome at both the protein and RNA binding sequence levels. Next slide. parCL, or photoactivatable ribonucleoside enhanced crosslinking, essentially takes conventional crosslinking one step further to increase the sensitivity and specificity of RNA-protein interaction capture. So while conventional crosslinking, or CCL, essentially crosslinks RNA to close proximity proteins at the 254 nanometer wavelength, parCL requires a pre-incubation step with the thiolated nucleotide, in our case 4-thiouracil or 4-TU, to allow this nucleotide to incorporate into nascently transcribed RNAs. This becomes critical because the thiol group on those nucleotides will be what specifically cross-links to zero-distant rather than close proximity proteins at the parCL specific wavelength of 365 nanometers. In either case, protein RNA complexes can be purified out, protein can be identified by mass spectrometry, and RNA can be sequenced. However, in the parCL conditions, you can actually also identify the RNA binding site on RNAs through mutational analysis, as any thiol nucleotide that was cross-linked will generate a T to C mutation. This is all dependent on pyrimidine salvage, which unfortunately Plasmodium falciparum does not inherently possess. Next slide. Lucky for us though, Heather has previously created a workaround for this issue by exogenously expressing a yeast fusion protein, FCU, that gives our transgenic 3D7 line the enzymes needed for pyrimidine salvage as well as parCL methods. Next slide. So after optimization of both 4-TU incubation and cross-linking conditions, we wanted to do a pilot analysis to see what kind of proteins we could cross-link and pull out with RNA. So focusing first on the left side of this slide, looking at a silver stain, particularly at our sample inputs, compared to a sample that saw no 4-TU. A sample that was incubated with 400 micromolar 4-TU had no dramatic differences in protein banding through silver staining, but also protein concentration. The same can be said for conditions of conventional cross-linking or CCL. and par-CL when compared to a no cross-linking or no CL sample, suggesting that together 4TU and cross-linking alone are not going to make any dramatic differences in the input proteome. Moving into the more exciting data, our purified eluents to see what kind of protein we're pulling out with RNA here. As you can see by silver staining, both the conventional cross-linking and par-CL samples have a lot more banding and more intense banding when compared to the no cross-linking, no CL control, suggesting that both of these methods are indeed working in our hands. I think it's also interesting to see between both cross-linking methods that there are differences in banding patterns and intensities, suggesting that each method may be able to bring out different RBPs with different strengths. Satisfied with this particular experiment, we took these eluent samples and submitted them to mass spec analysis in collaboration with the Indiana University Core. And we actually found 127 proteins enriched in our cross-linking samples when compared to a no cross-linking control. As you can see from the Venn diagram on the right side, the bulk of these hits were in our par-CL samples specifically. Of note, 19 of our hits were ribosomal proteins, and 18 of them have been previously annotated as RNA binding. Next slide. We wanted to first get a general idea of the function of the proteins we found enriched by cross-linking. So we did a Go-term analysis on these hits. And here I'm showing you any Go-term that was significantly enriched with the number of proteins for each term shown in each bar. And the results were pretty promising in that we're finding terms such as RNA binding, rRNA binding, structural constituent of a ribosome, and translation, all suggesting that functionally these proteins likely interact with RNA. Next slide, please. The next thing we wanted to do was compare our pilot study to the only other published in vivo cross-linking in Plasmodium falciparum done by the LaRoche lab in 2016. It is important to note there are some pretty big differences in methodologies here. The LaRoche lab looked at both trophozoite and schizont stages, where we looked at the trophozoite to schizont transitional stage. The LaRoche lab did conventional cross-linking and looked for RNA binding proteins that were specifically interacting with mRNA, whereas we did par-CL and conventional cross-linking and didn't do any particular RNA enrichment. So it's not surprising that their data set and our pilot data set have distinct differences that you can see by the Venn diagram on the right. But what I think is pretty exciting is that we do have some overlap, suggesting that our methods are able to pick up previously discovered RBPs in vivo. Next slide. So while that was exciting, there was so much difference in methodology that we also wanted to compare our data set to some bioinformatic analyses to see if we could pick up anything that's been putatively discussed as an RBP. So here we're comparing again to that same publication by the LaRoche lab. They did both in vivo and in silico analysis, but also we're comparing to a publication by the Lindner lab in 2015. The big distinction in their data sets is likely due to the fact that they're interrogating the entire proteome here, where we're focused on one single time point in the IDC. But nevertheless, again, we do see overlap, suggesting that we are capable of not only identifying in vivo discovered RBPs, but putative RBPs as well. What's a little intriguing is how many distinct hits we still have in our data set. And while it will take replication to truly confirm these as RBPs, it could suggest that we're picking up RBPs that may have RNA binding domains that haven't been interrogated by these publications, or just are non-canonical RNA binders. Next slide. So with the last few minutes here, I'd like to focus on our hits at a more specific gene level, starting with our previously reported RBPs that we found in our study. These are the hits that were outside of the ribosomal proteins. And what you can see is we're getting some of the big players, such as all three ALBA proteins, poly-A-binding protein 1, and some RNA modification enzymes. Next slide.
2-minute warning.
Next slide, please. I'm sorry. Thank you. And looking more on the potential novel candidates that we've found, we have been able to identify some candidates based off of domains that were not previously searched in Plasmodium falciparum, such as the WD repeat domain known to bind RNA, but also through function of orthologs in other organisms, such as HSP90 and different regulators of chromosome maintenance. Next slide. All in all, I think our study has established optimized conditions for PARCL in Plasmodium falciparum. Our pilot proteomic analysis identified both previously known and novel RBP candidates, confirming our method, but also suggesting the possibility of its increased sensitivity. Ongoing studies in our lab focus on replicating our mass spec analysis, but also optimizing RNA capture and sequencing to elucidate those RNA binding sites in specifically that transition between trophozoite and Schizont. Once we've characterized there, we'd like to extend our characterization throughout the IDC and transmission stages of parasitic development with an ultimate goal of understanding the RBPs critical for life cycle progression. Next slide. I'd just like to briefly thank my mentor, Heather Painter, my fellow lab mates, and our collaborators for all of their help. If you are interested in this research and would like to join our lab, we do have fellowship positions available. Please feel free to reach out. Thank you.
Thank you very much, Megan, for your presentation. We will take some questions now. We have one question in the chat. Individual malaria proteins with other functions, e.g. enzymes, are known to bind their RNA. Do you see that?
I'm sorry, let me just quick look at it. Yes, we do see some enzymes that have been annotated to bind RNA before. We do have a lot of uncharacterized proteins that are showing up as well. I don't know if we're catching everything since it is just a single stage right now, but I can confirm we're seeing enzymes that bind RNA in our hits.
Okay. So are there any RBPs that are currently being used as a drug target for malaria treatments?
Not that I'm aware of. I think there's been the thought tossed around of PUF2, but I don't know of anything that's gotten too far in development.